Welcome to the third day, and we're very fortunate to have Christine Kendziorski. I just asked her how to pronounce her name, and I stumbled over it. That's a little embarrassing. We're very fortunate to have her, not only because she's a internationally renowned statistical geneticist, but you should hear the problems she had getting here with all of the canceled flights and last-minute this and rescheduling. So we're very grateful to her for working so hard to get here to speak to us today. Thank you.
Okay. Thanks, Jeanette, and thanks for having me. When you live in LA, what I went through to get here might sound like a big hassle because you guys can fly everywhere direct, but I live in Madison, Wisconsin. So, 3 delays and we canceled your flight and put you through Dallas even though you're going to LA. This kind of thing is just second nature. So it was really no problem for me. I'm happy to be here. And I think I'm going to skip, because I had some problems getting here, you'll notice that I wasn't here for the 4:15 lecture, which I hope in a way, I hope we have time to get to. If we don't have time to get to it, I guess it means that people had a lot of questions and we had conversation, and that would be great. But I hope we have time to get to it. I think we will have some time, but I'm going to, instead of starting with it today, I'm going to start with methods for a number of reasons, unless there's some major protest in the room. So I'm skipping to the 9 a.m. talk. Yep.
Point the mic a little closer.
Okay. Oh, is it on?
I think it's on. It's just not turned up though.
Can you hear? Yeah, it's on. Got to be pretty close, I guess.
Yeah, it's going to be annoying.
Can you not hear me in the back if I'm not on the mic?
It's better.
A lot better. Okay. I'm going to do my best to stay put here. All right, so here's what we're going to start with, the 9 a.m. lecture, and I think I'm lecturing until noon, so the last lecture will be 4:15's lecture from yesterday anyway. So the methods that I'm going to talk about were motivated by a collaboration that I have with Jamie Thompson's lab at Wisconsin. And as many of you know, or might know, I think there's a mix of biologists and statistician backgrounds in here. The Thompson lab in 1998 developed a technique to isolate and grow human embryonic stem cells in cell culture, thereby establishing the first human embryonic stem cell line. So of course these cells are pluripotent, meaning they have the potential to become any cell in the body, and they've proven over the last almost 2 decades now to be incredibly useful for studies of basic biology, especially studies concerning development, and they're also a remarkably useful source of cells for regenerative medicine. Now, in spite of their utility, there's of course much political controversy surrounding human embryonic stem cells, and there's also concerns with compatibility, right? And so in an effort to, at least in part, address those challenges, in 2007, the Thompson lab published a method for reprogramming adult human skin cells back into a pluripotent state, and thereby established these so-called induced pluripotent stem cell lines, which are almost identical to human embryonic stem cell lines. Now, if they weren't almost identical, but indeed identical, they'd be ideal for research, right? Because their development doesn't involve human embryos. But they're not identical. There are some small differences. And so a focus in the Thompson lab, as well as many other labs across the country that use these cells, is characterizing those differences so that we understand their implications on downstream studies that use iPS cells instead of human embryonic stem cells. So to characterize those differences, the Thompson lab looks at all sorts of genomic information. They look at mutations between these 2 types of cell lines. They look at methylation patterns, protein, gene expression, and in particular isoform expression. So when we got involved with this project, what the Thompson lab asked us, this was a couple of years ago now, was can we accurately identify not just differentially expressed genes in an RNA-Seq experiment, but differentially expressed isoforms. Okay. So have you had background on RNA-Seq data collection? I'll go over this very briefly. So in an RNA-Seq experiment, what does a data collection look like? Well, you start with mRNA. Oftentimes the mRNA is fragmented and reverse transcribed to give cDNA fragments. These fragments are then sequenced. The sequenced reads look like this in the schematic, and we're going to see this later. come out of there, and they're aligned back to the genome or some reference transcriptome. And then this is the data that we use to get expression estimates. Once we have these estimates, we use these to identify differentially expressed genes, or in our case, to identify differentially expressed isoforms. So when we started looking at this problem, here's what the landscape looked like. There were lots of tools in place for each of those steps. For aligning reads back to the genome, discovering, de novo transcripts, for estimating expression, cufflinks, RSM, RNA-Seq was out there, and importantly for identifying differential expression, primarily at the gene level. Now, CuffDiff was out there to identify differential expression at the isoform level, but there were already 3 packages out there for identifying differential expression at the gene level, and we're not interested in making more work for ourselves, right? If there's packages out there and they work quite beautifully, even though I'm a statistician, I'm not super interested in coming up with the 4th or 5th method that improves mean squared error by a tiny bit and this kind of thing. So we thought that this was an easy problem, one that we would put in like the consulting category, where we have a new graduate student go and they run the 3 approaches and we compare results, and then we go on and do something that's a little harder. In fact, we found that that wasn't the case when we're looking for differential expression at the isoform level. These packages work pretty well, especially DESeq for identifying differential expression at the gene level, but not so great for identifying differential expression at the isoform level. So I'll spend some time today telling you why and sort of giving you the motivation for the approach that we developed. Okay. So all of these methods, just a second. All of these methods, and these are just the first few that were out there, and still anyone that you find to date, you'd be hard pressed to find a method for identifying differential expression at gene or isoform level that doesn't involve the assumption that counts are distributed for a gene G in a sample S as negative binomial with some shape and scale. So all of these methods assume that, right? I've got counts for gene G in sample S distributed as negative binomial with some shape and scale. And the shape and scale determine the mean and variance uniquely, so I'm just going to call those mean and variance, even though when you see a negative binomial distribution, you have R and Q, that's shape and scale, not mean and variance. But we'll call them mean and variance for practical purposes. The way in which these methods differ is in the way in which the variance is estimated. So, general idea, we make this assumption, and now before I can make any inference, like calculating p-values, I need estimates of these parameters for my data. And I'm going to estimate them by fitting this model to the data, right? And so the way these methods differ primarily is in how I get estimates of the variance. EdgeR assumes that the variance takes this form, and they fit this form to the data, and they estimate parameters. DESeq, slightly different form. BaySeq, slightly different form. Some of these methods share information across the genes or the I'll say the genes to estimate variance, and some of them don't. Okay, the problem, I'll give you this first. Well, now that I've said it, the problem is that with isoform expression, estimating that variance, there's some systematic differences in the way the variance is for different isoform groups. And if you don't account for that, then your inference isn't as good as it could be. So let me show you a picture of those differences. So again, here we have counts distributed as negative binomial, and I need estimates for the mean and variance. So what a lot of these methods do is some form of the following. They look at the empirical variance as a function of the empirical mean. So I've just taken a bunch of data here, and I'm plotting variance as a function of mean, and I fit a spline to that relationship. And then I say, okay, if the X bar for this gene across the samples is around 100, I'm going to use Well, this relationship changes with what I'm going to call isoform complexity. So you can't just do that when you're looking at isoforms. And what do I mean by isoform complexity? So let's come back to the picture that I showed you earlier. Here I have 2 genes, gene G and gene G prime, and here are the exons for those genes, okay? And gene G is really simple. It has one transcript that is transcribed during transcription. All the exons come together to make one isoform. Gene G prime is a little more complicated, depending on timing, depending on tissue, whatever. Gene G prime transcribes 2 different isoforms. Sometimes the first 3 come together to make a transcript, and sometimes E2 and E4 come together to make a transcript. So the number of transcripts that come off of a gene, I'm going to denote by NG. So NG in the simple case, the first case, always the same isoform, is 1, and here NG prime equals 2. All right. So the thing is, when we go back to the schematic of how mRNA data turns into sequencing counts, this is what I'm showing you. This is the data that I use to get expression estimates, right? And getting expression estimates is harder when you have what I'm calling more complex isoforms, isoforms where NG is bigger than 1. Why? Well, let's go back to this. So here's the data that I use to estimate expression, right? These are counts, these are reads that map to exons, and I want to estimate how many times this isoform has been transcribed. Well, I can just use all these counts and maybe take the average or the median or something like that. It's harder in this case because I don't know what to do with the counts that map to exons that are, present in more than one isoform, right? What do you do with exon 2's counts? The first methods just divided them up half and half. Okay, and then just applied standard approaches, mean or median or whatever. Then some approaches showed, okay, that's a big problem. Maybe I'm just going to throw them out. Well, throwing them out's a huge problem, right? Because most genes transcribe more than one isoform, so you lose a ton of data. What you want to do is you want to divvy them up in a way that's directly proportional to the expression of the isoform, but that's in fact the thing that you're trying to estimate. So it's this tricky problem unless you know something called the EM algorithm, which says, okay, if I knew the answer, this is how I would divvy these reads up. I don't know the answer, so I'm just going to iterate through until I converge on something. And that's exactly what our colleague Colin Dewey did when he developed RSEM. But the point I want to make here is that estimating expression here is harder than estimating expression here, and so your estimates of expression in these complex cases or more complex cases aren't as good. Another way to say that is that expression estimates for these complex isoforms, they have increased variability. And you need to account for that increased variability if you want to have accurate inference. So going back to the picture, check out what happens. Here's, if you just say, okay, I'm just going to fit the gene model to isoform data, right? And you're back at this transcript right here. It has mean 100, and you're going to use this to estimate the variance. Well, if it happened to come from a simple case of NG equals 1 isoforms, this is actually the relationship between the mean and the variance. So you end up overestimating the variance. You should be taking something from down here. Can't really see if I'm hitting that right, but you should be taking something from about right there, and you're taking it from somewhere smaller. I'm sorry, you should be taking something from the yellow line, and you're taking it from the red line, which is bigger. And here's the NG equals 2 isoforms. And here's the NG equals 3 isoforms, and you get the idea. So who cares, right? I mean, my graduate student, Ning Lang, who's awesome, plug for her, even though she already has a job, she came and showed this to me, and I thought, okay, that's pretty cool, that's exciting, but who really cares, right? I mean, we get the idea. Let's just assume all these test statistics basically look like t-tests. I assume everybody knows a t-test, right? We love the t-test. So I want to tell if there's differences between two groups. I'm going to calculate a mean over here in the first group, calculate a mean in the second group, look at the difference, and then normalize or adjust for the variance. And even when you do this fancy negative binomial modeling, whatever you're going to do, you end up with a test statistic that basically looks like a t-test statistic. You're looking at differences between groups normalized by some estimate of the variance. So if you use the red line to estimate the variance when you should have used the yellow line, you're using a variance that's too big, right? You're saying the variance is bigger than it is. So what happens to your test statistic? It's a quiz, so I can drink some coffee. Exactly, yeah, it becomes smaller, and so it's not as powerful as it should have been. So we got to worry about that, right? Those test statistics in general are underpowered. Even bigger problem probably in most studies although that's a big one because you don't want to be wasting your resources. If you use a variance, you should be using the pink or green estimate, but you're using the red line as an estimate. Now your variance is too small. That test statistic is artificially large, and so you have increased false discoveries in those groups. So you can get the idea of what might be going on, but again, are we going to devote 6 months, maybe a year, developing a nice method when we get 2% increase in power or 2% decrease in false discoveries? So we started looking at this to see what's the impact of this difference going to be. So here's the plot that I just showed you. This again, this is from Thompson Lab, and this is an estimate of the variance, just box plots of the variance for all the transcripts, and then for the simple ones, and then ng equals 2, and ng equals 3 contains the genes that have 3 or more isoforms, and you can see, okay, there's some pretty big differences, fine. And then we look to see, okay, is this just a Thompson Lab problem? Pretty good lab, but you got to check the lab, right? So here's some RSEM processed from another collaborator of ours. Maybe this is a Wisconsin problem, so we look at some publicly available data processed a different way. We see the same effect over and over again, another lab, cufflinks processed data. And so after seeing this, we did a bunch of simulations to see how much would it matter. Are we talking 1%, 2%, or are we talking 10 or 15%? And it ended up being the latter. So we were convinced that we needed an approach to deal with this, and that's what motivated this approach we call EBSeq. So here's how EBSeq works. So like everybody else, we don't want to be too unique. We too assume that counts conditional on some shape and scale, and again I'll call that mean and variance since the shape and scale determine the mean and variance, are distributed as negative binomial. So that's this, but what sort of a unique feature, I would say, of our approach is that we also put a prior on the scale parameter, and it's this prior that helps us deal with the differential variability. So let me tell you how this works in pictures and in thought, right? So the first thing we assume is that for a fixed R, so I'm just going to talk about Q as the mean, because I'm thinking of R as fixed here. We estimate it within gene. So some isoforms just have naturally low counts. Latent levels of expression are what I'm going to call them. We don't know what they are, and you can think about them as what you would measure if there was no biological variability and no technical variability. So if there was no biological and technical variability, we could make a lot more progress in science and we wouldn't need statisticians, right? So it's the true underlying level of expression for a given isoform in the tissue that you're looking at, and it's your target of interest. For some isoforms in the given condition, that mean is low, and for some isoforms, that mean is high. And there's natural variation from isoform to isoform. So this distribution right here governs variation in latent expression levels or true underlying expression levels. Okay. Now, conditional on a given mean for an isoform, now I've sampled that isoform. I'm looking at a particular one. It has its own latent level of expression. There's variability due to biological and technical variability. And so this level of the model says the counts that I see conditional on a specific latent mean and variance is given by this distribution right here. That's the negative binomial. So those are the two layers of the model. We can talk more about those assumptions later. I'll let you know that they can be checked, and we assume and recommend that you check them if you're going to use this package. But those are the two. Those are the two main components of the model, and I'll pause for questions on them before I go on. We're going to see this in detail in just a sec. This is in pictures. Mm-hmm.
Is it known a priori what the isoforms are?
Yeah, that's a great question. So when we started so the question is, do you assume that you know all the isoforms? And when we started EBSeq, we were working with known annotation. So we assumed we knew all the isoforms. We now have a sort of extension of EBSeq where you can, you don't have to know all the isoforms, but you do have to define uncertainty groups. So let's say you have some genome that hasn't been sequenced and you do de novo transcript detection and you have these estimates of expression for isoforms that are discovered, newly discovered. We have to group them by uncertainty, and there's a couple different ways that we can do that, and once we do that, then we can run EBSeq.
So in your first slide,
Mhm.
had the fourth exon only appeared in one of the transcripts.
Yeah.
And so the first thought would be, if you know that, couldn't you just use that one as a measure of the expression of that transcript? But E4 only appears in gene G prime.
Right.
Only appears in G prime 2.
Right. So the comment is, this one right here, this exon is unique to isoform 2, and I'm assuming known annotation, so I should know that this is the unique contributor, right? And so this, I will say two things. First of all, just to stress, EBSeq, what we developed, takes expression estimates as input. So you can use whatever method you like to estimate expression. You can use cufflinks, you can use RSEM, whatever you want, or you can use, we started out with RSEM and cufflinks, again, because we were assuming known annotation, but if you don't know the genome and you're going to do de novo transcript detection and estimation, you can use input from any of those approaches as well. So for our stuff, we kind of start at that level. We have expression estimates. But to get to your specific question, RSEM does exactly this. So in the model, if you look at the form of the estimates for these isoforms, the contributions from E4 will be upweighted because E4 is a unique contributor to that isoform. Yeah. Mm-hmm.
So when you have reads, you can also get exon-exon junctions.
Yeah.
Right? So in your model here, you're sort of only looking at transcript over just the exon. So how do you incorporate, for example, get a map read that spans exon 2 and exon 4?
Right. Right. So again, we don't, because we take output from methods that do. But in RSEM, so I've made this, so the comment is, in case you didn't hear in the back, this is super simple. Like, I don't talk about exon-exon junctions here, because they're hard to draw, and, you know, I made this a simple cartoon. But RSEM, the early stages of RSEM did not account for that, but they do account for that now. So they estimate, they have a step in here that I haven't shown you, where you get estimates of exon-exon boundaries, counts at exon-exon boundaries, and that helps in estimating isoform expression. But that, the method that they use would be, you know, specific to the expression estimation method, which again is input to our approach. Yeah. Mm-hmm.
For some of these isoform estimation methods, instead of a single value, it has a distribution, a component associated with it. Is it possible to incorporate that subtlety of isoform estimation into the model?
Okay, so I think there were two questions. So the comment was that a bunch of these approaches for identifying differential expression have uncertainty associated with them, and I'll talk about the uncertainty associated with posterior probabilities that EBSeq gives out. But then the second question is, can you incorporate the difference in uncertainty with sort of expression estimation? And that's sort of what motivated EBSeq, is that we noticed that methods developed for identifying differential expression at the gene level don't incorporate differential uncertainty at the isoform level. And so we do that. I'll show you the form. So in our model, we actually have different priors for different uncertainty groups. So this NG, beta NG, is different for different complexity groups. And we see more variability in the groups with more uncertainty. Okay, so here is the approach in equations, not in pictures now, so you'll recognize this one. X is the expression of isoform I, and I had a little GS that I'm dropping, but I want to stress it's specific to gene G in sample S. And P0 and P1 are the prior probabilities of being equivalently expressed and differentially expressed. So I just showed you the model, when I think about modeling data, I think about how would you simulate data from this process, right? If you can simulate data in a computer, you have a model for a process. Might not be a good one, but you have something. And I just showed you the model for data in a single condition, but now I'm interested in extending that model to 2 conditions, right? Because I want to know about differential expression across 2 or more conditions. And so P0's the prior probability of being equivalently expressed, and P1 of being differentially expressed. So this is just the form of the parameterization that we used. This is nothing new. Again, this conditional on R and Q within a condition. We have negative binomial, and I told you that R and Q determine the mean and variance. This is the way in which they determine the mean and variance. And so what I'm going to say, what I'm going to define as being equivalently expressed for a given isoform is if the mean, for a fixed R, if the mean is the same across conditions and it's differentially expressed, if that true latent level of expression that I talked about, the thing you don't know but you wish you knew, is different across conditions, then that's differential expression. Now, I don't know which case I'm in, so I'm going to say that the counts come out of some mixture model. There's some chance that they're differentially expressed, and there's some chance that they're equivalently expressed, and I don't know which case, so this is the form of the model that describes the data. Now, what does this F0 look like? Again, this is the model, the marginal predictive distribution that describes the counts in the event of equivalent expression, and it takes this form right here. So I integrate out this prior that I've put on, and this is the marginal predictive distribution under the alternative of differential expression, and you'll notice a couple of things about this approach. First, to get to the last question, here's the prior depends on NG group. So this is where we account for differential uncertainty. This form of this marginal predictive distribution is a beta negative binomial model, and here's its mean, and we're going to see that later on when we extend this to time course data, but in case you want to remember that. This is why I like mixture models. So when you have 2 conditions, you have 2 options, right? Those latent levels of expression are the same, in which case we say equivalently expressed, or they're different, in which case we say they're differentially expressed. Now, what if you have 3 conditions? How many options do you have? It's part 2 of the quiz. So with 2 conditions, there's 2 options. They're the same or different. But with 3 conditions, there are 5 options, right? No? Nobody likes that? So, so either the latent levels of expression are the same across those 3 conditions, or they're different. They're all different, or the first one is different than the second and third, or the second one is different than the first and third, or the third one is different than the first and second, right? That's what I mean by 5 options. You've got 3 conditions, and you're very interested, I'm talking a specific isoform measured in 3 conditions, and you're very interested in the differences and similarities of that isoform's expression level across those 3 conditions. You can have one of 5 options happening if we're not talking about ordered alternatives. You can worry about ordered alternatives later. We don't worry about that for reasons I can detail in case anybody's interested. But if you're not worried about ordered alternatives, you have 5 options. Everybody's the same, everybody's different, or the first guy's different, second condition's different, third condition. Okay, so if I have 5 options, then this becomes a 5-component mixture model. And I can evaluate the posterior probability of any option that I'm interested in just as easily as I can evaluate the posterior probability of each of these 2 options. So in our code and in our method, you know, any method you read says, and this just kills you, right, when you're trying to develop methods. In the abstract, it says, this can be extended naturally to more than 2 conditions. And so then reviewers are like, oh, that could be extended. Why didn't you compare to that? Well, extending naturally to more than 2 conditions is often super non-trivial, right? But in mixture models, it's actually not hard at all. I mean, it's a little, you got to deal with the code. But the method itself is not hard. For me to write down 3 more components here and evaluate, run through the same machinery to evaluate posterior probabilities of each of those patterns, it is quite natural, and we do that. So, so I like this kind of thing. Okay. Another thing for statistician types to notice and biologists to notice if they're worried about the assumptions of these models, This guy, we make the assumption of conditional independence. So our posterior predictive distribution under the alternative of differential expression is actually a product of the marginal predictive distributions under the null. So because this thing is a beta negative binomial, if you wanted to make other parametric assumptions and you could program this thing in, our code, it's just called a family in our code, and you can run all the machinery just like you run it for the parametric model that we've put down right here. So that's kind of nice if you want to program in other parametric assumptions, or you have a different type of data where the beta negative binomial doesn't fit very well, you can program in other assumptions. Finally, we will move off this slide, but I really like this one. We're all interested in the posterior probability of differential expression, right? That's what motivated this work. But in the context of mixture models, the posterior probability of equivalent expression has just as much weight, and you can interpret it in the same exact way you interpret posterior probabilities of differential expression. This is unlike p-values, right? Get a significant p-value, everybody's excited. If you don't have a significant p-value, what can you say? Can you say you're super sure that these levels of expression are the same? No, you can't say that with a p-value. But you can quantify the evidence in favor of equivalent expression with mixture models. And for this particular study, where we're trying to quantify the differences between human embryonic stem cells and induced pluripotent stem cells, being able to say something not just about where they're different, but also where we're really sure that they're the same was important. But I'll argue that it's important for other studies as well. Okay. So that's the whole basis of the method. Any questions before I go to simulations? Yep.
So does this assume that it's either one or the other?
Yes. Yeah, it assumes it's either one or the other. So for each isoform, you get two numbers and they sum to 1 if you have two conditions. And for each isoform, if you have 3 conditions, you get 5 numbers and they sum to 1. Yeah. Mm-hmm.
How much is the assumption of independence in estimating the differential expression affects your posterior probability?
So the question was, how much does this assumption of conditional independence affect your posterior probability? It affects it slightly, but not as much as you would think. So we have a paper from, I don't know, back in the 90s, where we evaluated this assumption, and what ends up happening is your posterior probabilities of differential expression, if there's a bunch of dependents, and we're assuming conditional independence, are slightly conservative, but you don't lose too much. If you ask that in a more aggressive way, if you had said independence, why would you assume everybody knows all the genes are connected, which lots of times we hear. And you didn't say that, so I'm not pointing at you, but just so everybody knows. I like to tell you what we assume. Every method assumes some sort of independence, just FYI. And if you know of a method that models dependence among all the genes, I would love to see it. And we would incorporate it. We would love to not make assumptions of conditional independence, but who can model dependent structure among all the genes? Maybe Steve can. But the rest of us humans cannot do that. For groups of genes, we do something like that. And that's sort of the simulations that we use to evaluate when you have predefined groups of genes, or what we use to see how bad this gets when we violate that assumption. Okay. So we do some simulation studies. And like everybody else, we assume, this doesn't bother anybody because everybody assumes this, so it shouldn't give advantage or disadvantage to any of the methods that we evaluate. For the set of simulations that I'll talk to you about briefly, we assume 90%, we have 2 sets. In the first set, it's easy, 90% are equivalently expressed. We simulate isoforms as well as genes, and 10% are differentially expressed. We assume the variance from edge R, so there might be a slight advantage given the edge R, but that's about it. And we take parameters and sample sizes from case study data to try to make the simulation pretty realistic. And then our second simulation is the same, except we take 10% and we put in outliers, because we, especially in the early days, tons of outliers. And we have a bunch of these. Okay. So first thing to check, do the simulations look at all like real data? So The means are easy to match, but the variances are harder to match from case study data because we don't have tons and tons of case study data. There's a lot of variability in estimates of the variance. So we check estimates of the variance. This is data from, a bunch of studies, and then our simulation is in light blue, so it looks okay. And here's some results that we see. So just looking up here, at the isoform level, again, cuff diff was the only thing out at the time and still is, specifically for transcript level, not gene level. And if you've used cuff diff, you know that you can get cuff diff and then on all of the transcripts, and then also something that they call OK, which is like transcripts that have passed their quality control filters, including filters for outliers, and so we run both. And we have substantially, increased power relative to cuff diff. And then the gene level simulation is comparable to these other approaches. So we can see some increased power, but not, I would say at the gene level things are comparable. And then here's false positive rate versus true positive rate for simulation studies, both at the isoform level and at the gene level. Okay, and then here is an evaluation of EBSeq is shown in red relative to these other approaches, cuff diff and cuff diff 2. Cuff diff 2, not recently, a couple years ago came out, I guess. And this is on the microarray quality consortium data where they have RT-PCR gold standard results on like 280 something genes. And so again, this is true positive rate versus false positive rate. And most of the gene level methods are comparable. EBSeq is a little bit better in the tail, but I don't think it's significantly better. Okay, so case studies. Again, this is the study that I talked to you earlier about that motivated this approach, where we're looking at human embryonic stem cells versus induced pluripotent stem cells. We have 4 and 4 in each group. This is repeated in triplicates. We actually have 12 and 12 in each group. I won't show you data today from the Gould lab, but in our paper, all the plots that we show for the Thompson lab are produced in the supplement for the Gould lab to show that we're seeing qualitatively similar results. So this was one of the first things that we looked at that motivated our approach, was a comparison of DE-seq versus EDGE-R for this study. And we can see EBSeq finds 90 genes. So of course we're really interested in ones that EBSeq finds that other methods don't find, and vice versa. And you can see here, these are the 144 genes identified by EDGE-R across the 12 groups. So these are human embryonic stem cells versus the IPS lines. This is 12 versus 12. And what do you notice here? I think we know, sorry, need more coffee. These are genes. These are samples, 12 versus 12. And these are the 144 genes identified by EDGE-R, and green's low and red's high. And what do you see? If you generated this, would you trust these results? What do you think?
Beautiful batch effect.
There is a beautiful batch effect. Do you think, then here's that beautiful batch effect, right? Check this guy out. What do you think the batch effect is causing the DE calls? Maybe, right? Hard to know for sure because depending on the resolution of the projector, these are slightly darker than these, right? So being called, clearly these are what I'll call batch effect. I like that. We've been calling them outliers. Whatever, outlier due to batch, okay? So clearly this is troubling. But if it's an outlier in the presence of a truly differentially expressed gene, I don't really care. That's fine, right? And again, these are slightly darker. So this might be true differential expression, or it might be a false call due to these guys. And then up here, this looks a little bit better, but you do see some outliers in there as well. Okay, this is what the calls from EBSeq look like. A little bit better, right? Here I'm a little bit more confident, but there's still this going on. And so we wanted to see what was going on. So what would you do if you were in love with mixture models and thought that they were really powerful, and you had this machinery and you wanted to check if these DE calls are truly DE or if they're due to outlier?
Sorry, I didn't catch it. Is that the one for isoforms
These are genes at this point. Yeah. So I'll show you what we did. This is EBSeq, and I'm just going to show you a picture of this model so that I can show you a picture of a bigger model. So this is the two-condition model. I have equivalent expression, differential expression. I don't know which case I'm in, so I have the two-component model, and I'm going to fit that model to the data and evaluate posterior probabilities of differential expression and equivalent expression. Here's a picture of equivalent expression, and here's a picture of differential expression. That's what I'm talking about. I've got condition 1, condition 2. If the colors are the same, the latent levels of expression are the same, and if the colors are different, the latent levels of expression are different. So we fit this model to the data. It's an 18-component mixture model, which you can do because you have 20,000 genes, and you don't have that many unknown parameters in this model, so you can get good estimates of them. What I have here are the 12 human embryonic stem cell lines and the 12 induced pluripotent stem cell lines. This is equivalent expression. That's one option. This is differential expression. These 8 patterns are equivalent expression with an outlier. So all the latent levels of expression are the same, except we see that batch effect outlier for these 3. Here's the batch effect outlier here, and so on. And then these guys, they're actually differential expression, but there's an outlier in there. So I can fit this model now to the data and evaluate posterior probabilities of each of these patterns. And I can see what's going on with those calls, right? And so I do that, and what I see is that for a bunch of these, there's a very high probability of being equivalently expressed with an outlier. And there were none of those extremely high probabilities for EBSeq. Now, truth in advertising, when I fit this model, I'm now assuming the model for EBSeq holds, okay? So EdgeR is at a slight disadvantage here. Even so, I can just look at data. So when in doubt, look at the data. So here's 9 genes that are called DE by EdgeR, but EE by EBSeq, and this is with pretty high posterior probability. And we can see what this looks like. These are 3 replicates at each of the 4 conditions. And this is probably a call due to an outlier, right? And we see this, again, maybe there's some action here, but I think it's pretty clear that you wouldn't spend all your farm money or whatever the thing. I wish I was better with those analogies. Sell the farm? I don't get it. My husband's sort of a farmer, and he's got all of these at his fingertips. Anyway, you wouldn't spend the farm? What is that thing? I don't know. Bet the farm. You wouldn't bet the farm on those. Okay. These are the ones that are called differentially expressed by EBSeq, but equivalently expressed by EdgeR. And so these look a little bit better. Some of these I wouldn't bet the farm on either, but relatively, there's a stronger DE signal here. Okay. And then again, in our work, and I'll argue in other work, a nice feature of this is that you can quantify equivalent expression just as you can quantify differential expression. So we were very interested in identifying differentially expressed isoforms that were in equivalently expressed genes. So if you just look at the gene level, you don't see any differences because there's compensation, so the averages look the same, but the isoforms that compose those genes or come off those genes are actually different. So we can pull these things out as well. Okay, so I think we're in good time for, what time do we break? Okay. Okay, so.
If you want to break, we have a break.
Okay, well, we'll break after the summary because it's a nice time to break. So the summary for EBSeq, methods for identifying DE in an RNA-Seq experiment need to accommodate the differential expression estimation uncertainty in different isoform groups, and that's what motivated EBSeq. It can be used to identify DE genes as well, even though it was developed for isoforms. It works pretty well for genes. It's robust to outliers. And I really like this feature that I've stressed, I think, is that it can be used to accommodate more than 2 conditions. So, with a frequency-based approach, if you have 3 conditions, you can move into like an ANOVA-style test from a t-test-style test, right? But when you reject with ANOVA, you don't know where the differences are. We say there's some differences among these 3 groups, but I don't have a good way, there are ways, but I don't have a good way to go in and quantify where those differences are, and EBSeq does that. So when we come back, I'll talk about extensions of EBSeq to time course data, and then we'll talk about single cell. Thanks.